All right, good afternoon, everyone. Let's finish up chapter 11, I think it is, the membranes chapter. Talking about integral membrane proteins, and we mentioned that you could do tests where you could figure out whether the N-terminus was on the outside or the inside. And the reason why you'd want to know that, well, you need to know which part of the protein is functional to the outside. And it's very helpful because some proteins will have the different termini on the inside or outside, and we even call those type 1 or type 2. So notice that the inside is on the top of this figure. I don't particularly like that about this figure, but type 1 has the N-terminus on the outside, and that's sort of what we would think of us going from outside the cell and we're reading the protein. Maybe we think of that as normal, and so we call that type 1. Flipped on its head to have the C-terminus facing outside, that's type 2. And then you have a couple of things. The only important ones are type 3 and type 5. It's a little bit weird. Type 3 is where you have one chain that crosses the membrane several times. Both type 1 and type 2 only have one membrane crossing region. Type 3 has multiple regions. Type 5 has multiple regions, but they're in different chains. So you can just think of them as being sort of like increasing numbers of complexity in the sense of number of chains as you go up. And there's no type 4 because type 4 is given to something that I don't tell you to remember. It's that weird kind of anchored protein. So basically just worry about the ones that we have in the box and just know the difference between these four types. For example, I could show you Bacteriorhodopsin right here, and it's a type 3 integral membrane protein, clearly one chain, and crossing the membrane several times. It doesn't matter, but the amino terminus is outside, so if it was only crossing one time, it would be a type 1. But just follow the rules and know how to name the different things. And this is interesting because if you think about it, rhodopsin is the protein that lets you see. It's in your eyes, and so I would expect it to be sort of floating around inside the cell, but actually no, it's actually a membrane protein, and there's a reason for that. And we're going to talk about that when we get to chapter 12. But the main thing is, it's a pretty big membrane protein, and it's not just incidentally peripherally on the membrane, it is integrally integrated into the membrane. And this actually works by opening a door for protons to cross the membrane. Notice also, this is in bacteria. So our version of rhodopsin is going to be a little bit different, but it's going to look pretty much like this. So it's kind of interesting to think about the differences and the similarities. Bacteria don't have eyes, but they have a rhodopsin. And rhodopsin is a membrane protein that lets protons through. So one of the things you could tell is you could sort of take a guess, even from the primary structure, that Bacteriorhodopsin is a type 3 protein. Because you could do what's called a hydropathy plot. And this is like what we do in the lab, where we just look at the residues and we add up a window of residue properties and we say, oh, that property is really high. And they add up whether it's hydrophobic or not. They call it the hydropathy index, okay? But the main thing is you slide down the protein and you go from the first residue to the last, and you see that you have regions that are more hydropathic, they're more hydrophobic than other regions, and they tend to be about the same width, exactly what they should be if these were 7 membrane crossing helices. So rhodopsins have 7 membrane crossing helices that all fold up into, they snake back and forth across the membrane. If you look at this, and I just want you to just pause for a second and look at this, and if I gave you a test question saying, okay, which type of protein could this be on a hydropathy plot? Take a look at that. And if you look at it, there's only one region that is about the same length. It's about 20 or so residues, and they're all hydrophobic, and that sure looks like a membrane crossing alpha helix. There's only one of them. And so you can conclude it's not type 3, it's probably not type 5, but you can't tell whether it's type 1 or type 2 without more experiments. So the other thing you can do is you can look at proteins in cross-section, and you can label them different ways. Here's 5 different proteins that are all integral membrane proteins. And just notice there's some interesting patterns here. We colored charged residues blue, tryptophans red, and tyrosines yellow. And if you notice, the middle of each of the proteins is pretty gray. You know, there's not many charged residues, tryptophans, or tyrosines. The blue charged residues are located both on the outside and the inside of the membrane. Not many inside the membrane itself, a couple that just have to break the rules, but remember, none of these rules are 100% rules, they're like 90% rules. The other thing is, look where the tryptophans and tyrosines are. Those are interesting because they are located sort of around where the head groups are located, at the interface between water and lipid. So you have the charged residues outside the membrane, and also inside the cell, but outside the membrane. You have tyrosine and tryptophan at the water-lipid interface, and inside you have neither. So uncharged residues are located inside the membrane. And what's interesting is that you can even have a structure of the membrane that lets it move. Remember that these integral membrane proteins, if they're not anchored to something else, they'll be able to sort of move around the membrane like this, right? And what's interesting is that they found out a certain shape will be like what they call an icebreaker. It will actually be sort of a wedge that allows the protein to cut through the membrane and move faster. Accelerated diffusion, because they actually distort the lipids and it makes them more unstable in the region of that protein. So you can have certain proteins that are like fast movers within the membrane. And I just think this is kind of cool. How does this work? We're just figuring it out right now. But there can be shapes of proteins that can distort the membrane and cause the protein to diffuse faster than the other ones who just sort of diffuse more slowly. Just notice it has this weird protrusive shape. It's something in the shape of the protein itself that disrupts the shape of the membrane. And the other thing is, we talked before about the sphingosine-based lipids are a little bit thicker than other lipids. They're a little bit longer. The sphingosine chain, I believe, is more like a C18 chain than a C16 when you line it all up. And that means that since sphingosines will self-associate and they're taller, you'll get these little things, and this is a scanning microscope thing of the thickness of a membrane. And you'll see that you have these thick regions where there's little islands that are floating around that tend to have a lot of sphingosines in them, and they tend to be thick. And these tend to have different chemistries as well. You can have different proteins that are even anchored with lipids that will associate with these little microdomains. And so it's not just proteins floating around the membrane. You can have microdomains of thicker proteins that are themselves floating around the membrane. Here's a cross-section of a microdomain, and you can see there may be additional cholesterol, but there may be sphingolipids in particular will be enriched in these thicker microdomains. And they'll look at this from the side, and it's just kind of cool. It's another way in which the membrane is like a mosaic. It has these little thick bits of lipids that go around, as well as thick proteins that sort of go through the membrane. And these will be able to float around. They're even able to rotate a little bit. And here's a thing about how when you have a thicker microdomain, it sort of tilts to one side a little bit, and that makes it tilted so that it actually forms a little bit of a vortex. And how does this actually affect how the membrane works? We don't know, but it's really cool to look at right now. And you can see that different temperatures will result in different kinds of vortexes. Here's a particular vortex. This will spin faster when it's at 28 degrees Celsius than it will spin when it's at 21 degrees Celsius. And so why does this happen? How does it affect how things work? We're just figuring this out right now. The other thing that you might have done is you might have in the lab have done electroporation. This is where you open up holes in a bacterium by literally zapping it with a pulse of electricity. And so you do this to get a plasmid in and out. It's one of the ways that you can get a plasmid into the cell. And we now know that the electroporation works because it literally rips holes in the cell. But because the membrane is fluid, the holes will close back up as long as you don't shock it too much. Sometimes you can fry your sample, but usually we have it worked out. We can zap it just enough to make it like Swiss cheese. And this is a computer simulation of what happens when you zap. When you zap, these holes open up. Those are big enough for a plasmid to cross through, but then the membrane will flow to fill in those holes in a short time, a couple of seconds, and the cell will be happy again. A little stressed, but happy again. And there's other ways you can make holes in a membrane a permanent hole if you have a protein that goes through the membrane. And look at the structure of this protein. That you're looking at it from the sides. The membrane's going back and forth. And if you look at the gray protein, it's all beta sheets. And in fact, it's a circular beta sheet that wraps around on itself, and it makes a big hole in the middle. And if you peek really closely at that, you can see there's a whole protein in red. The red thing is another protein that is literally passing through the membrane hole made by this beta barrel. I think that's pretty cool. So you can make pretty big pores if you make a big beta barrel, and that can be what the cell needs to let entire proteins pass through. There's a number of different ways beta barrels can be used, and here's just 5 different examples. You can do lots of things with beta barrels. The one thing I want you to look at most is the one all the way on the left. There's a side view of the Hemolysin toxin. I say on the left, I mean on the right. Go all the way to the right, look at the Hemolysin toxin. And what it has is it has a big hole that it punches in. And look at the name, hemo, blood, lysin, breaking. This is a blood-breaking toxin. It's made by another organism that wants to blow up your red blood cells, and it literally will have a beta barrel that punches holes in the membrane. Once those holes are punched, then basically the ions will flow in and out until they reach equilibrium, and a cell at equilibrium is no longer alive. A cell at equilibrium is dead. And this is exactly how hemolysin actually works. It's a soluble protein in part A that's a monomer. When it reaches the cell membrane, that little orange part flips down and it inserts into the membrane, and it gets together with 6 other of its buddies to make a heptamer, a 7-membered heptamer, a 7-membered oligomeric complex. Okay, let's say it's got quaternary structure, but it forms its beta barrel from 7 of those beta hairpins associating together to make a big hole in the membrane. I think that's pretty amazing how this protein works, but I certainly don't want it getting anywhere near my own red blood cells. I like them very much intact. Thank you very much. Here's another beta barrel that was shown on a previous one, and it's a big beta barrel. And they show it letting a protein through here, and it also is letting water through. They show the waters as the blue blocks on the left side of this. So I think it's really cool that we can do that. The final thing that you'll see is that these are, of course, embedded in the membrane, and the bond goes both ways. These are stuck deep into the membrane, and if you take them out of the membrane and put detergent on them and put them into crystal structures, and you get the crystal structure, you will see that there's a layer of lipids still attached to the protein that are stuck so tightly that it's difficult to rip them out. And you can literally see these frozen in place. These are called annular lipids. They are stuck so tightly to the protein, they're practically a part of it. And this is an example of an aquaporin. This is a pore membrane protein that lets water through. It regulates the water concentrations inside and out of the cell. When you get the crystal structure of this, on the top you actually can see the aquaporin. It just needs to have a little hole to let water molecules through. And you can kind of see that the annular lipids form sort of an imprint of the membrane. You can see the blue head groups and the yellow chains, and you can see kind of where the bilayer was there before the rest of it was ripped away. The one on the bottom is the FO protein, another one that you'll talk about in Biochem 2, and it is filled with lipids. You can see the little bilayer sort of stuck on there. You can't scrape it off because it's stuck so tight. And that leads us to the last topic, which is what in the world is that cardiolipin doing? Here's a picture of cardiolipin, and it's like 2 phospholipids stuck together with a glycerol literally connecting the 2 phosphate groups. So why would you need something like this? Well, it turns out that this is used as annular lipids that stick tightly to membrane proteins, and it's used to tie 2 membrane proteins together. You find it in the ATP synthase, which is a big membrane protein structure, and the mitochondria want those to be located in the same part of the membrane, so they staple them together with cardiolipins. One end sticks to one protein, the other end sticks to the other, and they literally tie the proteins together by being annular lipids that have covalent bonds that are linking and bridging the protein complexes. I think that's pretty amazing that cardiolipin works like that, and it means that these complexes are really important, and it's really important to the cell that they stay together. You'll be talking about why in Biochem 2. But all that's enough for Biochem 1. So this finishes Chapter 11. And make sure that you move on to Chapter 12 sometime this weekend so that we're ready to continue Chapter 12 on Monday.